Medical School Histology Basic. Today we're going to talk about skin or the integumentary system. We want to talk about the diversity of skin and what structures of skin, cells of skin that make possible that functional diversity. Thank you. Medical School Histology Basics, Integumentary System or Skin. Hi, I'm Larry Johnson. I'm a professor at Texas A&M University, and today we're going to talk about skin. Our objectives is to gain a greater appreciation for the diversity of functions of skin and to recognize the different cell types and structures of skin that make possible this functional diversity. Now, what is the function of skin? It protects us against injury. You see this little boy has injured his leg. It also prevents against desiccation. We are terrestrial beings. We live on the land. We don't live in the sea because we do not desiccate. And our skin maintains that, maintains water balance, excretes substance, thermal regulations. It receives temperature, pain, as that little boy is experiencing, and pressure. Also, skin is a basis of recognition, and it yields clues about one's well-being. You can look at these two ladies and tell right off which one is the daughter and which one is the mother. Also, fat metabolism in the subcutaneous or hypodermis layer, this fat here, is an energy source for metabolism. Now, skin has a binary origin. There's three origins: ectoderm, mesoderm, and endoderm. And two of those is where we get skin. Ectoderm is the epidermis, and the dermis come from the mesoderm. From in here, we can see this in relatively thin skin. Here again, we can see the dermis, which has the fibroblasts and a lot of collagen materials. And then we have the epidermis, which is the epithelial cells. Stratified squamous epithelium with keratinocytes on the surface. Now, there is regional variation in the epidermis. It could be thick, like a sole. It could be thin, like the eyelid or most of the body. Here we see it fairly thin in the ear. It could be transparent, like in the cornea of the eye. You can see right through it. Also, it has appendages. Appendages could be a nail. It could be glands. And also, it has projections in the oral cavity whenever you have the tongue, for example. So it gives rise to numerous appendages, hair, as I mentioned. Also, the mammary gland is epidermis or originated structure. Nails, as we said. Apical sweat gland, apical sweat gland as well, but this is one that marks the territory in animals. And then you have the tarsal gland, which is right up here at the top and at the bottom. It holds the tears in. And the tears themselves, all those come from the epithelium of the epidermis. Now, if we look at thin skin, and it's thin because the epidermis is thin, as opposed to thick skin, thick because the epidermis is thick, we can see a host of things. We can see the different layers: the epidermis, the dermis, and the subcutaneous layer here. Epidermis is thicker here, dermis, and then the fat layers. We can also see in skin the hair follicles, sebaceous glands that we were talking about, blood vessels that project up into here. Epithelium sits on the basement membrane, and the basement membrane separates the epidermis from the dermis, as we can see. So when you pull the epidermis off of the dermis, you see in thin skin versus thick skin, it's really the epidermis that determines whether it's thin skin or thick skin. And you have thick skin like a sole of the foot or on the hand. You ever worked in the garden, you might get a callus. You're developing thicker skin in that region. As opposed to your arm or the belly or someplace, you have thin skin. And I just want to illustrate here that you can use your atlas. The atlas has things labeled for you and identify the different structures in there. the arteries, the veins, the sebaceous glands, the sweat glands, all those can be seen in the atlas images. Now, if we look at abdominal skin, which least specialized and most prototypic of skin, we see the dermis is dense, irregular connective tissue, and the epidermis is above. If we look at thicker skin, we see a thicker epidermis, dermis, hypodermis below, and you can see where the epidermis has pulled off. And whenever this happens, you have the interdigitation. Thicker skin has a greater interdigitation between the dermis and the epidermis than does thin skin. And here we can see that projections of the dermis into the epidermis we call dermal papillae. And then the epidermal pegs, or the reedy pegs, are the epidermis dipping into the dermis itself. So where the epidermis touches the dermis, we call that the papillary region, where you see these projections in through there. And then below that, the dense irregular connective tissue is the reticular layer. Papillary layer, reticular layer. And here we can see the papillary layer projecting up through there. This would be like a sweat gland duct going out through there. And if you look inside these projections, what you will see is blood vessels. So these are the blood vessels that provide the epidermis, which is avascular, with its nutrients. Here we can see the blood vessels here, which are the same as what we're seeing here, which are in the dermal papillae. In addition to the dermal papillae projecting up through there, you can even have a corpuscle. This is a Meissner's corpuscle that is light touch sensor that we have. Dermal papillae, epidermal pegs, epidermis, dermis, as we can see. And then the hypodermis, which is below the reticular layer, below the papillary layer, which is the interaction between the epidermis and the dermis. We have the hypodermis, which is where the fat cells are associated with them. Also, there's a proscenium corpuscles. You see these onion-like things in through there. And then we have an eccrine sweat gland. So this is the one that makes you sweat throughout the body, is the gland that does that. And here's a duct of it. The duct is more eosinophilic or red as opposed to the secretory portion. So we have fat cells, adipocytes, and the hypodermis, and this is a deep pressure detector in through there. So there's a proscenium corpuscle, sweat gland ducts, DUCT, and also the sweat gland itself, the eccrine sweat gland, and another proscenium corpuscle, which looks like an onion, but these are like fibroblast cells here, and a little piece of a nerve is in the middle there. So there are different layers of the epidermis. Palms or sole of feet is where we see those very plainly. You have the stratum corneum on the outside, which is dead cells, differentiated cells are divide here and to differentiate as they go to the surface. And this is the most differentiated cells. The cells are flat, dead, nucleated. You don't have nucleus in them. You just basically have keratin hyaline granules. Below that, you can have the stratum lucidum, especially in thick skin, which is just a clear area through there. And then below that is a stratum granulosum, and this is where you see the keratin hyaline granules, which is the matrix of these cells up through there. And then below that is a stratum spinosum, where you have spines in between here. Looks like spines. It's really desmosomal connections that hold the cells together, and they interact with the tonal filaments or the intermediate filaments. And then the very bottom layer is a stratum germinativum or stratum basale, and that's the source of mitotic activity that then differentiates and moves to the surface and ultimately is sloughed off. So we can see those in real skin. This is thick skin with the keratinized epithelium, keratinized because it has a stratum corneum on it. Stratum corneum, stratum lucidum is right in through there. Stratum granulosum is where you see the granules that you see through there. Stratum spinosum, you can see the little spines in through there, which are desmosomal connections between adjacent cells. And then the basale. The basale is the basal layer that actually sits on the basement membrane that interacts with the dermis, and it has hemodesmosomes as opposed to desmosomes connect two cells. Hemodesmosome connect the epithelial cell to underlying connective tissue that we see there. So this is the keratin hyaline granules, and this is keratinized stratum corneum, as we can see. So you can have a little clear stratum lucidum, stratum granulosum, stratum spinosum, and stratum basale. And here we can see the similarity to the keratin hyaline granules here, and we can see the desmosomal connections of the stratum spinosum, hence their name spinosum. Basically what it is, the space should not be there, but when the cells shrink, the desmosomes between them hold tight. And so you see the spines. Really, there should not be a space in between there. So basically, 4 layers: stratum corneum, stratum granulosum, stratum spinosum, and stratum basale. If we look at the stratum basale, we see hemodesmosomes. This is collagen in through here, and this is the basal lamina or the basement membrane that all epithelium sits on. Stratum spinosum, you can see the spine. These are the desmosomal connections between this cell and that cell, this cell and that cell, and they look like little spines when the tissue shrinks. So this region here is what we're seeing around these cells, which is a stratum spinosum located between the basale and the stratum granulosum. And the stratum granulosum has a keratin hyaline granules, and that's what we see at the light microscopic level. We can see these granules, and above that is a stratum corneum. Now, from the base, all the way from the basal all the way to here, you have desmosomal connections. You see them very plainly here in the stratum spinosum, but they continue all the way up here, even in the stratum corneum. You can see where the cells are attached to one another with the desmosomes. Now, the matrix of these are just basically the keratin hyaline granules. The nucleus has been removed, the cytoplasm is removed, the cells differentiated dead, and it just sits at the top, and ultimately they're sloughed off. And they're sloughed off the top, the stratum corneum, stratum granulosum. You see the granules in through here, spinosum, and the basale. If we look at electron microscopic level, you can see how these cells are packed in through there, stratum corneum, stratum granulosum. You see the keratin hyaline granules, which is the same as what we see there. And then this is a spinosum, and then the basale is at the very bottom layer. We don't have one of those. So these guys are keratocytes, which are the main cell type that divides at the bottom and differentiates as it goes up to the top. There are some other cells in there. One of those are Langerhans cells, and you can see the dendritic processes of the Langerhans cell, and that's associated with the immunologic barrier, detecting things come through there and telling the immune system. So there are different cells. The main cell that comes through here is the keratinocyte that goes in through there. Keratinocyte, the main cell type, from the ectoderm. As we see, there's also melanocytes that it's from the neural crest, and that's associated with pigmentation. The Langerhans cells we just saw, which is in the spinosum, immunologic role, and then the Merker cell is a cell that interacts with the nerve endings, and so it helps detect things for the nerve endings. Here we see the melanocyte. It too is a dendritic, namely it has branches, as we see here, and it's located in the stratum basale. And so here we can see one of those cells. There's a cell, and these are dendritic processes. This is stained for the enzymes that are in the melanosomes. But if we saw that in a regular piece of tissue, this is the basale, and these clear cells here are the melanocytes, because they do not make desmosomal connections, and as a consequence they shrink as opposed to being attached to the cells, and that makes a clear cell that you see. The Langerhans cell is a clear cell that's located in the spinosum. So if you look at the melanocyte, it produces the melanin granules, but it passes on to the keratinocytes, which has it. So here we see the melanocytes, and this is where the epidermis is being pulled off of the dermis, and we're just looking at these ridges here in the epidermis because the melanocyte is at the very bottom of the epidermis because it actually touches the dermis as well. So this is epidermis with projections of the melanocytes that we saw a while ago. And so the melanocytes are clear cells that produce at the bottom, and then it passes on to the keratinocytes, and the keratinocytes on the base, the ones on the base here, put those melanin granules, you see there's kind of golden granules, above the nucleus. So when the sun's coming down through the stratum corneum, stratum granulosus, spinosum, and stratum basale, the DNA inside here is protected by the melanosomes that are there. And here we can see the capping, we call that capping, and here you can see the melanosomes, the capping of those cells. Now whenever the melanin granules are not evenly distributed, that's whenever you get freckles to occur. If you don't have melanin, if you fail to produce that, that's albinoism. That's the rats that we see is albino. The red that we see there is really blood that we see. Malignant melanosome, the cancer, we don't want it. And then Addison disease, also pigment distribution. So you have different pigmentation associated with adrenocortical insufficiency. So there are some diseases associated with the melanocyte, and this is not malignant melanoma is not a good one to have. Here we see the epidermis and the dermis interaction, and these piles that are produced here are due to the fact that there's more cells around the basal here than there are between there, and so these cells pack up, and that makes a little ridge, and in fact that's what makes your fingerprints. And if you look at this ridge is different from that ridge, is different from that ridge. So if these ridges on the same finger are different, of course, a person's fingers would be different from one another and also different from other people. So that's the basis of fingerprinting. There's also appendages. You got the hair follicle. This is a rector pili muscle that pulls the hair up. If you've ever seen a cat that's upset with the hair sticking up, it's the rector pili muscle that pulls it up. If you had chill bumps or cold bumps, the rector pili muscle is pulling this portion of the dermis, and it makes a little indentation as it's trying to pull the hair follicle up. So this is the dermis, but you have the projection of the hair follicle down into the dermis, as we see there, and this is the subcutaneous layer or the hypodermis that you can see. Here we see hair follicles projecting up through there. Sebaceous glands discharge in the hair follicle, and that's what makes your hair kind of oily. And a pili-sebaceous unit is a hair follicle, sebaceous unit, and a muscle, the rector pili muscle. Here you can see the rector pili muscle, sebaceous glands, and right in here would be where the hair would be located in through there. Now, if you have straight hair, you have straight follicles. But if you have curly hair, you'll have curly follicles. And here you can see the curly follicles. That's going to yield curly hair for this individual. So we see the sebaceous gland that's going to dump into the hair follicle, and that's what the oily stuff that makes your hair oily over time. This is the rector pili muscle. So the hair follicle, sebaceous gland, as well as the rector pili muscle, make the pili-sebaceous unit. So there'd be one here, one there, one there, one there, one there, different units. And the sebaceous gland has holocrine secretion, where the cell itself is a secretion. That's why you don't see an lumen, because these are the secretions in the lumen. In addition to sebaceous gland, you have an eccrine sweat gland. This is the one that makes us sweat, which lands on your fingerprints, or also the cooling of the body. So this is the glandular portion, and then this is the duct portion of it. So you have a glandular portion and a duct. So this is a gland, and the duct is more eosinophilic, as we can see there. And when it comes through thick skin, you can see where the opening of the duct has to go all the way up to the top, because remember, it's actually discharged on the surface of these. You can see the sweat gland duct going up through there. Now at the recto-anal junction, you may have hair follicles down through sebaceous gland, eccrine sweat glands, but here you can see that junction. This is simple columnar epithelial cells with goblet cells that you see in the large intestine, and then this is the anal region here, stratified squamous epithelium. So you go from simple columnar to stratified squamous epithelium, and at this location is where you have these apicrine sweat glands. The apicrine sweat glands are bigger lumen than you see the eccrine sweat glands, and these actually discharge in the hair follicle itself. So there are different mechanisms for release of secretory products. You have MEROCRINE secretion, which is really just exocytosis. And then you have apicrine secretion, where you have some loss of the plasma membrane there. And both of these are in milk. So casein comes out through MEROCRINE secretion, but the fat, in order for the fat to be solubilized, it has to have a little membrane around it. So apicrine secretion, a little bit of membrane comes loose with the product. And that's in contrast to holocrine secretion, which is sebaceous glands have a holocrine secretion. The whole cell goes, and the cell itself is the secretion, where here milk fat is a secretion, or casein. Here the whole cell is a secretion. And the eccrine sweat gland that is for cooling is on the surface of skin, but the apicrine sweat gland discharges in the hair follicle itself, and the sebaceous gland discharge in the hair follicle too. So you can see those. And the sweat glands, the eccrine sweat gland, discharge on that little ridge, and that's why you have the fingerprints, because these little ridges here, you leave your fingerprints on the ridges. Also, the sweat glands have myoepithelial cells, and the myoepithelium forms a mesh around the secretory unit. And here you can see where it's kind of cut longitudinally, and you can see that the pink cells are not continuous. They're just intermittent as they form a basket around them. Contraction of that squeezes the sweat out. Here you can see the myoepithelium, how they make a net around the secretory unit and squeeze that. We can see again those cells. There's different types of granules in skin, and those are important in the function of skin. One is the myelin granule, and that's the one associated with, produced by the melanocytes and passed on to the keratinocytes, and that's the one associated with UV light reduction on the basal cells. Then there's a membrane coating granule. Here we see a membrane coating granule, and that's located right in the stratum granulosum layer, and that cements up between the cells, and that's what prevents desiccation. And then we have the keratin granules, where we see in the stratum granulosum, and that's really the matrix of the cells, which are really mostly tonofilaments that you have. And so it's a matrix of those that we see there, but small membrane coating granules seal this. And so you can put your hand in water and then back out, put it in gasoline, put it back out. I wouldn't recommend it, but you can. And you don't have leakage because of the sealing of this between adjacent cells. And so the keratin granules is what's the matrix up to there. It's really insoluble. The keratin is insoluble, has a lot of disulfide bonds in there, which makes it very, very insoluble. So you can see the keratin granules, the melanosomes that we see. Now, in terms of regeneration of the epidermis, if the part is removed, still has sweat glands, and still has hair follicles below it, then it can regenerate the surface from these cells. In fact, that's the different degrees of burn. First degree, second degree, third degree has to do with how deep this is cut. If you cut it below the epithelium source, you'll have to have a skin graft in the third degree burn. Now, the dermis, below the epidermis, is a network of collagen bundles, and it could be muscle. Here we see some smooth muscle in through there. And we have smooth muscle at different locations, the nipple, penis, also the scrotum. And this is actually the scrotum here. This is the epidermis, very thin, and this is the dermis, very thin skin. And the dermis has smooth muscle, as I mentioned. The rectopili muscle is a smooth muscle, and this is the sebaceous unit, the pili-sebaceous unit here, hair follicle, sebaceous gland, rectopili muscle. And it has skeletal muscle for facial expressions. Hair follicles, gland, blood vessels, nerves abound in the dermis, because the dermis is a source of the nutrients to feed the epidermis. Epidermis is avascular, so it needs to get all its nutrients from below. And for facial expressions, different expressions that you can see that the people have, all these expressions have to do with skeletal muscle, and you can control the skeletal muscle. Now, some stratified squamous epithelium on the outside is keratinized, that is, have a very thick stratum corneum. However, if it's in a wet surface, and this is the lip, this is a muscle here, but this is a lip, it's non-keratinized. In other words, instead of having stratum corneum in through there, the nuclei of the cells go all the way to the top. Normally, in keratinized epithelium on the outside, the outer layer, stratum corneum, the cells are dead, but not so in the non-keratinized. And here we see a sheep, a lip. And so here we see hair follicles. So this is keratinized epithelium here. And then right in through there, it changes. And so here you can see non-keratinized, non-keratinized up here, which is going into the oral cavity. And you can see nuclei all the way to the surface. But this is a keratinized, and you see a thick stratum corneum located in here and here. So the epithelium could be, stratified squamous epithelium, skin, could be, it could be keratinized, located in through there all the way up to there, or it could be non-keratinized if it's associated with the oral cavity. And we can see that in the tongue. If you look at it, it's mostly non-keratinized. There are certain places, the filiform, if you've ever been licked by a cow or by a cat, you know that they had to have some keratinization to be such a rough surface in there. And here we see the tongue of a monkey. And we see the filiform papillae projecting up through there. And you can use the atlas to see the fungiform, the filiform. This is the one that doesn't have taste buds. And it's associated with moving the food under the teeth, which is the purpose of one of the purposes of the tongue. Now, when you have the epidermis and the dermis together, you have the different senses. You got heat, cold, pain, touch, pressure, and there's some nerve endings associated with that. We saw the Meissner's corpuscle that we saw here, which is in the dermal papillae. And then we saw the Purcinian corpuscle, which was down low, deep, more in the hypodermis that we saw. So in terms of the clinical correlation, albinoism associated with failure to make tyrosine, which is a tyrosine is a protein that's important in making melanin. Here we see a peacock, that's an albino. So we want to thank the many sources of which I've taken pictures from. We did not generate these. These are from these pictures. We want to thank the original sources for many of those pictures that we see here. So in summary, skin has diverse function, many functions, and it protects you against desiccation. It's a mechanism of recognition or one's well-being, detects pain, stores fat, and we have some questions about the integumentary system. Which layer of the epidermis has meiotic activity? The answer is none. The basal layer has mitotic, but none of them have meiotic. This occurs in germ cells. So this would have mitosis, but not meiosis. None of these would have meiosis. Now, which of the following skin granule function matches? Melanin, skin pigment? Yes. Membrane coating granules, waterproofing? Yes. Keratin hyaline granule forms a junction between adjacent cells? No. It doesn't form a junction between adjacent cells. It's A and B. Which function of skin involves granules? Protects against UV light? True. Protects against mechanical injury? True. Protects against desiccation? True. All those functions is what it has. Then the dermis has muscle, a skeletal muscle, and smooth muscle. Yes. Has elastic fibers that are reduced with age. Yes. The elastic fibers are replaced with collagen, and so it's less elastic. That's why we have wrinkles in older age. Has a reticular layer that interacts in direct contact with the epidermis. No, it is not the reticular layer. It's the dermal papillae that actually interact with the epidermis. So in the last 15 years, I promoted STEM in over 35,000 middle and high school students and their teachers. And our website, puretamu.edu, was hit 8 million times last year alone. So that's the end of medical school histology basic integumentary system. I'm Larry Johnson, and thank you for your attention.